Hello everyone, it's my pleasure to introduce a multiplex interactome of Ebola virus proteins reveals TM9SF2 as a cell surface attachment factor that promotes viral entry. Ebola virus was first discovered in 1976 and causes severe hemorrhagic fever with a high death rate of 50 to 90%. It has caused repeated outbreaks in Africa, including major epidemics in West Africa and the Democratic Republic of Congo. The virus relies on interactions between its proteins and host cell factors to replicate and disrupt normal cellular functions, though many of these mechanisms are still not fully understood. Highlights. First, we constructed a comprehensive EBOV-host PPI network consists of 1,728 core high-confidence interactions. Second, TM9SF2 was identified as a crucial host attachment factor that facilitates EBOV pseudovirus infection, both in vitro and in vivo. Third, targeting TM9SF2 significantly impairs EBOV pseudovirus infection, offering a promising strategy for antiviral intervention. We constructed an EBOV human interactome using two complementary approaches, based APMS and proximity labeling. For the APMS experiments, each of the 9 EBOV proteins was individually expressed in cells and protein complexes were purified in biological triplicates. For proximity labeling, biotinylated proteins were enriched using antibiotin antibody in triplicates, and analyzed by MS. We built a high-confidence EBOV human PPI network consisting of 1,728 core interactions. 3 complementary methods effectively captured low-abundance interactors and distinct functional enrichments. The core interactome contained 25 kinases, 13 transcription factors, and 201 known drug targets, underscoring the importance of multi-method integration for systematic dissection of virus-host interactions and antiviral target discovery. To investigate host factors involved in viral entry, we classified core GP interactors by cellular component annotations. This revealed that 304 core interactors localized to membrane-associated organelles. TM9SF2 was identified in multiple omics studies and linked to infections by various viruses, suggesting a potential role in viral entry. However, its function in viral infections remains unclear, and its involvement in EBOV infection has not been previously established. Loss of TM9SF2 significantly impaired EBOV pseudovirus infection, and re-expression of TM9SF2 in knockout cells restored susceptibility to pseudovirus infection. These findings established TM9SF2 as an essential host cofactor for viral entry mediated by EBOV glycoproteins. TM9SF2 knockout impaired replication-competent RVSV Ebola infectivity, surface binding, and internalization, with re-expression fully restoring these defects. These results identify TM9SF2 as a host factor that promotes viral binding and internalization during early entry. Biochemical assays confirmed a specific interaction between the extracellular domain of TM9SF2 and the RBD at the plasma membrane. TM9SF2 knockout reduced RBD binding, which was rescued by re-expression. Blocking TM9SF2 with antibodies dose-dependently inhibited RVSV Ebola infection. Thus, TM9SF2 is a novel cell surface attachment factor for EBOV that promotes viral binding via RBD association, highlighting its therapeutic potential. TM9SF2 heterozygous knockout mice challenged with EBOV pseudovirus showed significantly reduced bioluminescent signals versus wild type, particularly in females. Thus, TM9SF2 haploinsufficiency impairs EBOV pseudovirus entry and propagation, supporting TM9SF2 as a critical attachment factor and therapeutic target. Summary: this study provides a comprehensive analysis of the EBOV human protein-protein interaction network. Identifies TM9SF2 as a critical host factor that promotes viral attachment and internalization during EBOV GP-mediated pseudovirus entry. Targeting TM9SF2 represents a promising therapeutic strategy for antiviral intervention. These findings advance our understanding of EBOV entry mechanisms and pave the way for developing host-targeted therapies against EBOV and other viral infections.